folks from the Institute for Next Generation Healthcare for having me here. Late of 2016, one of my patients came to me, and he's a gentleman who had well-documented chronic and neurologic Lyme disease. He had been around the block a few times before he came to me. He had gotten a couple of courses of IV, and he would get better, and then he would relapse. And under my care, we designed a regimen that pretty much kept him well with what I call industrial strength amoxicillin, minocycline, and Malarone, because he also had babesiosis. And he's one of those patients that many of you probably like him. They're always scanning the horizon for the next best kind of treatment. He had been following the work of Kim Lewis for a while. You probably knew about Kim Lewis, maybe not quite as long as I had been aware of Kim's work. And because of that, he probably ferreted out that there was this YouTube video of Kim Lewis giving the keynote speaker address at the first annual Mount Sinai School of Medicine, Lyme in the Ear Precision Medicine back in October of 16. At the time, I wasn't even, for some reason, I wasn't even aware of that symposium, but he found this video on YouTube, and this is a photograph of mine of Kim at one of the LDA conferences in Philadelphia, not the Mount Sinai one, but during that keynote speech, towards the end of the keynote speech, Kim mentioned that in the test tube, disulfuram was more or less, sort of the best, most potent agent that had been identified, bar none. And he referenced the foundational work of Jayakumar Rajadas and Ravi Potaneni, who published their study with high throughput testing in vitro and identified disulfuram as very, very highly active. This is Dr. Potaneni. He was the lead author on that. And Dr. Rajadas, who's going to be speaking shortly. And in case you're not familiar with it, this is the drug disulfuram, which has a very, very complex pharmacokinetics. And so my patient saw this, and he approached me, and he's a very respectful patient, very trustworthy, and he says to me, "Hey doc, can I try this stuff?" So, first of all, I thought about it. Here's an FDA-approved drug. It's got a long track record. It's been in use for 67 years for the treatment of alcoholism. And at that point, you know how much I know about disulfuram? 0. Because I never had occasion to use it. So I did a little bit of research on it and familiarized myself a little bit about it and kind of figured out what's the toxicities from it, what's the dosages. And but before I agreed to let him use it, I want to pose this question: like, why would I grant his request? Why would I grant his request? And I don't have time to go into this in detail. And that's the other thing. I've asked the folks from LymeMind to post all of these slides because I'm not going to have time to go over them in detail. So this is going to be, I should have said at the beginning, this is my mission impossible, which I've chosen to accept, which is to tell you all about disulfuram and the treatment of Lyme and babesiosis in less than 15 minutes. But some of the things I say and a lot of the things I don't have time to say are on my slides, and I would refer you to look at those afterwards because I'm not going to be able to cover everything. So why would I consider granting his request? So one of my famous patients, Vicki Logan, was a patient who had, it's a long story, I don't have time to go into it, but she turned out that she was treated in a way that was supposed to cure Lyme disease, and then it turns out later on the Lyme organism was grown from her cerebrospinal fluid at the Centers for Disease Control in Fort Collins, Colorado. This is the abstract that was submitted at the 5th International Congress on Lyme borreliosis in Arlington, Virginia. And you'll notice that the co-authors include folks from CDC. And the conclusion was culture of this cerebrospinal fluid specimen of VSK2, that's a culture media, yielded a strain of B. burgdorferi, culture-confirmed treatment failures have been previously reported for 3 Lyme neuroborreliosis cases in Europe. The present case, apparently, is the first of this type to be reported from the United States. So this made the front page of the Science Times and New York Times in August of 1993. And this is me at Vicky's bedside in Northern Westchester Hospital Center. And this also resulted in a guest commentary in the Journal of Clinical Microbiology. I love this quotation from Charcot of the Salpêtrière. Disease is very old, and nothing about it has changed. It is we who change as we learn to recognize what was formerly imperceptible. And this was from that paper. This is back in the early 90s. Chronic persisting infection not yielding to antibiotic treatment presents a dilemma for the patient, the physician, and for insurance companies that are contractually obliged, or should be, to pay for medically necessary treatment. The solution is not the denial of the reality of patient illness or imposition of arbitrary restrictions on allowable durations of treatment, but the design of more effective and less costly treatments that can keep patients well. Aside from the prevention of the illness in the first place, methods achieving sure cure for those already infected must be developed. Antibiotics may not be the answer. Rather, application of new techniques of molecular biology to interfere irreversibly with key metabolic or reproductive processes of the bacterium, wherever it may be found in the body, including intracellular sites, may provide more effective and targeted therapy. So I was ready. I was ready for better treatments or other options. The Logan case and several other cases were printed in my article on chronic Lyme and the spectrum of antibiotic responsive chronic meningoencephalomyelitis. And by the way, this article is now available online. If you go to the Disulfuram article that is published under MDPI antibiotics, there are supplementary links where you can access this article in depth. And it discusses the Logan case. I don't have time to go into this, but she ended up developing a pleuropericarditis after she was out at the Mayo Clinic for implantation of a baclofen pump, which didn't really work out. But while she was out there, the rheumatologist there, because she did have some markers of autoimmunity, decided that she might have lupus and put her on big slugs of steroids, and then shipped her back to me, and she developed this huge pleuropericardial effusion. So this is her mediastinum, and it's mostly fluid. The heart is right in the center. And with that kind of condition, you often have to create what's called a pericardial window to allow the fluid to escape so the person doesn't develop tamponade. But anyway, this is her pericardium. And this is a touch prep of her pericardium by Paul Duray, who was at the NIH, showing a beautiful Borrelia-compatible structure. This is after she had gotten even more antibiotics. So in other words, proof that we're not curing the infection with our available antibiotics. I'm going to rush through this. This is another patient of mine who had a grapefruit-sized rash on his thigh. His wife brought them to the doctor. The doctor didn't have any idea what it wrote. This was in the mid-'80s. And then he tested negative for Lyme disease, so the wife kept telling to people, could he have Lyme? They said no, because he was seronegative. It's a long story. It's all reported in that article, which I encourage you to see. He developed massive hydrocephalus, and he had a florid meningoencephalitis and cerebellitis. And his tissues were sent to Dagmar Hlinska at the Borrelia Reference Laboratory in the Czech Republic, who on electron microscopy identified structures that she felt were Borrelia, and also with primers from Ben Luft at Stony Brook, she was able to detect signal for B. burgdorferi. So what I'm getting at, these are my own patients, studied in depth, proving persistent infection despite very intensive antibiotic therapy. So that's why I was open to trying something different. Let's just sort of repeat the same thing, that we need better methods of treatment. Okay, so let me get back to this patient. So anyway, I agreed to let this patient be treated, and whenever he had got off antimicrobial agents, he deteriorated within a couple of weeks. I said, you know, if we're going to do this, I want you to stop all your other medications, because otherwise I wouldn't know what disulfuram did or didn't do. I prescribed disulfuram for him, and I kind of picked a number out of a hat just based on what the usual doses were for treating alcoholism. Sort of picked the upper end of the standard dose, which is 500 milligrams a day, and I said, I want you to, he lived at some distance, so I said, send me a note every couple of weeks, let me know how you're doing. I want you to have labs every couple of weeks, and we did all of that. His labs were fine. And then a couple of months later, he leaves a message on my office voicemail, canceling his follow-up appointment and declaring, I'm cured. And he happened to mention in passing that he had required a hospitalization, but he didn't tell me why. So naturally, I'm curious, right? So I call him back up, and he tells me that he's feeling well, he's off all of his medication. And I said, well, what did you need hospitalization for? And then he mentioned to me that he had required a psychiatric hospitalization. And then I did a little bit more in-depth reading about disulfuram. It turns out disulfuram can affect neurotransmitters, and there have been some reports of neuropsychiatric illness, even frank psychosis related to the drug. So I called him back, and I discussed it with him, and I pointed it out to him, and he says to me, Doc, I don't think it was the disulfuram, because he was under a lot of situational stress at the time. But then he says to me, but even if it was worth it. And don't fail to offer that to other patients just because of that. He was like practically admonishing me over the telephone. Anyway, he's now 27 months out on nothing, more than 27 months, feeling well, and we've been in touch with him on a regular basis. So, I mean, that just kind of knocked my socks off. I've been in this field for 30 years. We've never seen anything like this. After that experience, I shared it with a couple of other patients, and again, I don't have time to go into it. It's all outlined in excruciating detail in my article, but patient 2, patient 3, similar experience. And these were very tough cases. And I said, after the third patient, you know, I said, I got to report this experience. So that led me to reporting this case, these cases and this experience with disulfuram. So, so that's the article. It's open access, and again, there are supplementals so that you can link to other information. Okay, so I don't have time to go into all of this. There's going to be slides. Let me just say that the optimal use, the optimal regimens for the use of this drug are not completely defined and remain to be defined. This agent, in my experience, is extremely potent. And although I started the first 3 patients just arbitrarily on a fairly hefty dose, which, by the way, I wouldn't recommend for most people, and nowadays we're starting at very, very tiny doses, like 125 milligrams every 3rd day only, and slowly ratcheting it up, and that way it's much more tolerable. And what I've been finding is some of the patients that I started on this drug were very, very precarious in their condition, and we started them on tiny doses, and finding that even 125 milligrams every 4th day. One of my patients is a 260-pound gentleman, who was very, very ill. And these patients started making incredible progress. So I don't know whether that kind of low dose will enable you to achieve a, quote, enduring remission or not, but it's very therapeutic. There's lots of stuff that I'm going to have to skip over just because of time. I've only got a couple of minutes left, right? This is, I don't want to call this a guideline, heaven forbid, it's just my protocol. There was a time when Kahn's current therapy would describe the method of prominent physicians in their field, and that was quite fine before the year of guidelines. So let's just call it the method that I'm using, and it's subject to change, okay? And this is going to be available on the LymeMind site. And there's some patients who have needed to be retreated, and we're still figuring this out. And another thing to say, we don't even really know at this point what the mechanisms are by which this drug has its effect. And it's a very complex drug and has a very complex pharmacokinetics. I'm just going to skip to this. Unfortunately, I have to confess, I do not know how to do Excel. So on my office nurse and I just put this little graph together, and this is our. People want to know this, so I want to get this out. This is our summary of the first 30 patients on Disulfuram. And probably 85% of the patients endorsed deriving benefit from application of this treatment. And then where you see those little E's in that column toward the middle, those are the patients who are enjoying what I'm calling enduring remission, meaning they've been able to be off all treatment for at least 6 months, remaining well. A lot of the other patients I haven't followed long enough to know that they're going to be enjoying that enduring remission, but I also want to indicate that we've had some significant adverse reactions to this drug. So this is a tricky drug that really requires care, titration, and the patients really need to be followed carefully. Some of the patients have developed peripheral neuropathy, which seems to be different than the Lyme neuropathy, although it's been pointed out it's sometimes difficult to distinguish peripheral neuropathy from Lyme. Anyway, and we've had a few patients who've had some emotional upset, nothing serious, but you have to really follow the patients carefully. And also, one important thing is to know when to quit this agent. I just want to, there's a lot of unanswered questions. Also, what's the mechanism? I just want to bring this up to you. Vicki Logan ended up passing away, and if you want to know the details of that, you can find that in my book or my 42-page letter to my then Congressman Chris Gibson, which explains what happened to her. But her tissues have been studied, and in fact, Eva Shapi just published with colleagues a detailed study of her tissues, and her tissues are rife with biofilm. Every cut of every tissue, brain, heart, liver, kidney, and not just small amounts of biofilm, large biofilm aggregates up to 300 microns, laced with beryllia. If you know anything about biofilm, it has a structure, there's a metabolism, oxygen is going in and carbon dioxide is going out. One of my speculations about one of the reasons why Disulfuram might be more effective than our standard antibiotics, one of the breakdown products of Disulfuram is carbon disulfide. So just remember the periodic tables. Guys, remember that? Sulfur is directly below oxygen. So what I'm getting at is carbon dioxide is a small molecule, very diffusible. Carbon disulfide is a very small molecule, very diffusible. It's known that carbon disulfide has antibacterial properties. So my pet theory, but I really don't know, and it really deserves further studies, one of the reasons that Disulfuram may be effective is that this breakdown product, carbon disulfide, may be able to diffuse widely, including in biofilm. Anyway, thank you all for your attention. I think is my time up?